Hi, thank you everyone for joining, and thank you so much for registering to this live webinar. Special thanks to those who are joining us from regions where it's very, very late or it's very, very early. And I wanted to present this webinar for our March series in 2016. My name is Baljit Ayubi, and I will be hosting this webinar on behalf of the Early Career Members Network. And as I mentioned, this is the March webinar series, and it's brought to you by the Early Career Members Network Committee. Today's speaker is going to be Reza Salek, who I'll present in a few slides' time. So the mission of the EMN, the Early Career Members Network, is to really provide a forum for metabolomics researchers at the start of their career and to serve the early members of the Metabolomics Society. And so the idea is to strengthen communication, collaboration, encourage opportunities and inventions, and to support development learning and to really enjoy professional growth throughout. And so the vision really of the committee is to establish a real sense of community and engagement between our early career researchers globally through initiative and enthused partition. So we have a new committee this year, of which I'm one of the members. These are some of our other committee members, some people you might recognize. And if you ever need to contact us, you can use the email address at the bottom of the screen there, which is info.emn@metabolomicsociety.org. One quick reminder that I wanted to bring up before our talk begins this morning. As you're probably all aware, the Metabolomics Society conference is just around the corner. It's happening in June from the 27th to the 30th this year. The location is in Dublin in Ireland, and so they will be in Europe this year. And just wanted to bring to your attention that the abstract submission is actually open. However, the deadline is March 30th. So you have a couple more days to get those poster and oral abstracts in. Please do go to the website, which is metabolomics2016.org. There are a couple of other key dates there on the side around registration dates. But what I wanted to highlight to you is what the EMN is doing. And we have a couple of different items on our agenda. We have a social mixer, which will be on the Sunday evening before the conference starts. That's just to introduce ourselves, to meet more of the early members of the Metabolomics Society. We're also conducting 2 workshops at the Metabolomics Conference, and these are listed here. They'll be on the first day, which is the Monday, 27th, at these times. And then there's also an EMN reception at the conference, which is after the poster session on Tuesday, 28th of June. And we'll also be chairing a young researchers scientist talks on the Wednesday of the conference. All of this information is up on the metabolomics2016.org website, and I highly encourage you to go there and take a look. So today, to go to today's speaker, I'm proud and really pleased to announce that Dr. Reza Salek is speaking about how close we are to metabolomics, reproducibility, data sharing, data standards, and workflows for metabolomics. Reza joined the EMBL EBI while remaining as an honorary research fellow. He was involved in establishing and promoting metabolites, which was really the first general purpose metabolomics repository. And he also helped manage and coordinate COSMOS, which is the coordination of standards in metabolomics. It's a large EU infrastructure project on metabolomics standards. He serves on the board of directors for the Metabolomics Society, chairs the Data Standards Task Group. He's also a chair of the Website and Communication Committee, amongst other task groups too. And so he's been involved in a variety of joint grant applications, journal publications, editorials and book chapters, and he's actually organized and run many different national and international courses as well as workshops. And so now I want to hand over to Reza. Take it away, Reza.
Thank you, Bao, and thank you, EMN, for inviting. The title is very grand, so a lot of things about reproducibility, data sharing, standards, and workflows, so probably each one by itself stands as an independent talk by itself. Obviously, this is a lot of work of a lot of people. So a lot of people work in data standards and create workflows and data sharing. So it's a whole team behind of these things. So I just want to acknowledge at first, and whenever there is a slide, I try to acknowledge that this is also a work of a lot of people. So if I miss someone and I forgot to mention them, I apologize in advance for forgetting to mention their name and acknowledging the valued contribution to all these efforts. Where I come from is EBI, so there's a place that I'm spending most of my time and work there. This is part of EMBL, European Molecular Biology Lab, which handles a lot of computational databases and resources. It's where you want to sort of say how good we are. We compare ourselves to CERN because it's modeled after CERN, but essentially works on dealing with data. It's a very international institute, and different nationalities work in there, about 500 people. Not all of them are bioinformaticians, but vast majority are. This is sort of an overview of all the services. There are a lot of services. Historically, when it started dealing with partly my mouse works in this area of genome, nucleotide, functional genome, and going down towards protein, proteome interaction, protein sequence such as UniProt and Prime. There are other resources deal with literature and text mining. There are those structure protein families like Interpro, PDBE, which is sort of molecular structure biology, down to small molecules and pathways. So this is where we sit, the group on chemoinformatics and metabolism. One of the services is Metabolite. That's the one mainly. I'll be showing some slides here, which deals with metabolomic experiments. But we have got other services that deal with pathways and set up. Reactome is the very well-known one, which deals with human pathway mechanism. So all in all, the mission of EBI is to handle data, to make this data reusable, available, to extract knowledge and provide a platform so people can access data, can reuse the data, can do research on the data. Historically, a lot of other DNA-based genome, microarray, and proteome are fairly more established. I think Metabolite one is a new addition, relatively new addition to this. So we're still learning and progressing, and sort of this is what I'm going to try to share with you. Because we're dealing with a lot of data and handling data across EBI, it is important for the data to be standardized, be in a format that could be accessed, used, available, and follow certain structural guidelines. You can't just share your data any way you feel like, because you might understand the data. But to make it available, accessible, and for other people to reuse this data, you probably have to go through a set of structures, a framework, a standard, a set of tags, and make this data machine readable, computer accessible. So for data to be used, and there are initiatives, standard initiatives across different fields. It helps and provide a guideline. A set of guidelines say that you need to provide a set of minimum information, that it is at least minimum information that I think you could share with me that is enough for me to reproduce the results. So the term minimum information could be misleading. It's as little information as possible. That's not the case. It is as enough information. That makes the data reusable and reproducible. So I can take this set of information, and there's enough information for me to go and redo the experiments or reuse the data. So every resource is involved in certain initiative, from PSI to proteome to functional genomic standards, and for metabolomics will be Metabolomics Standard Initiative, or MSI, which provide a set of guidelines to follow. Just give some numbers of how many people access their resources. These are for the geeks among you who like different data. So there are about 18.5 million requests every day from web services. They estimate about 5 million scientists use EBI resources, and these are cross resources. And as well as jobs. These are requests sent, about 9.2 million jobs, and about 60 petabytes of storage across EBI. Excuse me. So why we need to do that? I mean, these are two examples I thought just demonstrate the fact that obviously all of you have heard we are going to a digital revolution. We're creating data on and knowledge on a really, really fast and exponential rates. The estimates being, for example, by 2020, every 12 hours we are doubling the knowledge. And it will be very difficult to handle by ourselves. We need tools, we need machines to be able to handle this knowledge and exponential growth of knowledge and help us with assisting and analyzing data, and that's why we use computers. But computers cannot just use it yet without making this knowledge available in a format they could read, they could interact, and they could make a meaning of it. So the whole purpose now on will be how can I make this data machine readable, machine accessible, so actually it could help me to give me a structure to this data, and later on how would I be able to get this information back if I'm a different person and reuse this data. To do that, you have to go through certain rules, certain tasks, certain regulations that needs to be established to be able to knowledge mine the data. So we have all seen the publication medium. So we all try to publish and perish or make more publications, but these are existing classical form of sharing data as a form of a journal. You've been doing that for many years. This is not something new, but you might realize that sometimes it's not enough to get the result. You read the paper, you follow exactly what it says, but you actually can't reproduce the result. So it would have been good to actually have access to the data or more access to the protocols they use. Then your chances will improve. But also this data needs to be in a format that is accessible. Maybe it's on a commercial tool and you might not have access to commercial tool to be able to read the data, to access the data, to improve you understanding the result and making sense of it. So it needs to be in a format that it could be exchanged and changed to. There are also rules that always comes up. Always people are worried about the data. There needs to be clarification. Maybe there's medical data, there are copyrighted data, so there are ethical and privacy data. So all of these needs to challenge and address, and we are learning as we're going to make it portals for these data to become available. And there is a big data being the new black, which means the new oil of the future. So there are a lot of people talking about set of terminology, making the FAIR principles, and I think these are really good principles. We at Metabolomics community should apply and should use that. These are to make the data findable, accessible, interoperable, and reusable. So we need to make our data available. It has to be accessed. It could be used beyond just using metabolomic, maybe interoperable, meaning you might do a multi-omics data. So you want to be able to use data from metabolomics and combine it with proteomics or transcriptome and reuse that. So these are a set of slides and guidelines. I don't have time to go into the details of it, but if you follow the link for data FAIR port and data share and FAIR, you will have lots of information about this kind of principles of data sharing. So would that help? This is a good example. A good example being a software program. This is done by Kristen Kohlberg and Todd Propsting. These are in the US and Arizona State. It's a computer program. So the idea is if I share my code and say my algorithm, this is not biology, this is data already in a computer readable format by machines, how reproducible this will be. So he started from asking 600 people to do, and as the line goes through, there are exit lines, which are drop-offs, or in red, where it did not manage to reproduce the result. So there are different reasons. There are theoretical reasons that the data, although in a digital format, they could have not run it because maybe they didn't have the right hardware. Some people just were not interested. And some people said, you're not going to do it. So those who followed, which are over here, if you can see my mouse hovering over articles, web, and emails, after people said, I can use the article, or I can use the web information, or they start asking about my emails, what about this type parameter? How do you do that? How do you do it? So out of 600, about 231 managed to say, oh, we got enough information to go and now reproduce the result. But as you can see, almost half failed and half succeeded. So even though this is extremely good success rate, so if I say 1/6 was successful to reproduce a result, which is a format, an algorithm, a software, which is already in a digital form. And they were not very pleased with it, but if I put that in context of biology, this is really good. I think the success rate in biology will be far, far lower than that and far more challenging. Be able to get this many people and go through a reproducibility process. So it's a lot of challenges to have that in consideration. Going back, what set of guidelines? These are historical link. It's very nice summarized by Reza in the Metabolomics publication. You've got the reference link below. I think the title is called Water, Water Everywhere. And it summarized all the working group with the Metabolomics Standard Initiative, which they started and provide these guidelines for working in biology context. What do you need to share in terms of reporting in a paper? What type of chemical analysis you do? How do you report that? Or how do you tell how you process your data? And there are stuff about data exchange, what format should be used to make this data available, and also something called ontology. I'll get to that in later in more details. So these guidelines end up being something about 14, 15, or number of publication around 2007. There were updates to these publications around 2010 and 11. It is kind of asking that this is the best way to practice at the time we understood. But the problem is that it's a lot easier to say, you do this, you have to follow this, to write the guideline, but actually you want to do it, it's a lot harder. Always when you have meetings, we ask people, okay, well, how do you like this to be done? And other people, you have to talk about this, you need to report that, you need to talk about this thing. This is really important and a key thing to be shared or to be told about. And these are absolutely right. They're 100% correct. But when we ask the question back, okay, are you now willing to sit down and produce all of these things and share it with us? The question goes, oh, no, it's too much work. We're not going to do so. There's a practicality element to it. The low-hanging fruits are always easier. So here's where the real application of this MSI comes in terms of a database, a resource that will say, okay, we take this minimum information, we take these guidelines, we provide a medium to be able to capture these things in a digital format, a file format. I will show that again later. It's called ISOTAB. And to capture this information, this is not the only way. There are other ways, and there are other resources who use different things. For now, we have two major resources. So Metabolite started earlier, and then later on Metabolomics Workbench, which is an NIH funded. So obviously there are a lot of databases and resources related to metabolomics, but these two are the main one which are very global. So they accept any kind of metabolomic experiment. There has been in the past specialized ones. For example, they only accept data for NMR, and if it's only a plant, or do accept GCMS if it's done in plant. So such resources exist, but the two over here, which is metabolites from EBI, if you follow the address below, you will be able to see it, and Metabolomics Workbench, which will provide a comprehensive resource for all detailed metabolomics experiments for you to go and share and make it available, and each follow certain formats. So I'm going to more talk about our formats. But before, there's also another resource that came out of our standard initiative. This is called Metabolome Exchange. This is a kind of aggregator. Excuse me. This follows if you type in Google metabolomexchange.org, you will reach that. It's sort of, if I oversimplify it or simplify it and say this is kind of like a Google of resources, at least for repositories. So rather than going and search for your experiments in Metabolomics or go in Metabolomics Workbench, you can actually use this and type it there. There are resources, Metabolite, There is Metabolomics repository in Bordeaux, Metabolomics Workbench, and Goldman there. So if you search, for example, something like Arabidopsis, you will find all the data sets related to Arabidopsis that are example could be found. In total, about 33, some in Metabolite and some on Workbench. So it provides a quick way and fast way to go and find this data sets. So here now we're going to be a bit experimental and engage you guys in it. I know these topics could be a bit tedious, so we do a bit quick poll and see how much you're familiar with these things. So if you can launch the poll and anytime you can, then we could sort of ask a question, engage you guys with it, so at least you won't fall asleep from me talking continuously. So Bao, can you launch the poll now, please?
So the question is, how long would it take to share your experimental data, average complexity? Select one of the following.
So it is, imagine this is not too complex, not too simple. Average complexity, maybe about 100 samples. What is your gut feelings, essentially? If you haven't done this before, what would it take for you to complete and share this metadata? I haven't told you yet how it's been done, but let's see your answers. What is your gut feeling of how long would it take to share an average 100 sample metabolomics data?
Okay, we've got 70, almost 80% participation right now. Reza, do you want me to show the answers, or do you want to wait till later to talk about it?
No, please show the answer.
Okay. I'm going to close the poll in 3, 2, 1. And let me get my window back to share the results from that. All right, everybody should see the results.
Okay.
Do you see it, Reza?
Yes.
Okay.
So I have to say the couple of hours, people, I have to disappoint you. It's unless it's your second time or third time, you will not be able to do a couple of hours. It probably takes a lot longer. So those who you got about a day or two or about a week, you are right. This usually takes for the first time user to average do it. And if it's overcomplicated data, usually if it's parts are missing, then hardly goes about. If you've got a very, very complex study with intervention studies, thousands of samples, then yes, it could take a month. And no idea, people, you're absolutely right. So can we go to the next one, please?
Sure. Okay, here we go.
Okay, because I keep mentioning MSI, and this wants to give a feeling for me how much of you or participants here are familiar with metabolomics. I mean, these are sort of abstract questions. You might not be even working in metabolomics, so there's no expectation to know about MSI, but just want to know the gut feeling of we keep trying and see how many people are familiar, heard about MSI. So this will be an update for me to see how many out of the audience here are familiar with Metabolomics Standard Initiative, or used it, or using it in the publication, or using it in the reporting, or just heard about it but never used it.
Okay, we have 84% participation, so I'm going to close the poll in 3, 2, 1. And then I'll share the findings. There you go.
Okay, this is I think is a very good result. Being the 15% of you who said you using it is very good. I've seen a couple of papers which actually they start reporting the metabolite identified with the MSI levels. I showed it as well. I think the familiarity is growing, that people who heard about it never used it, but people who are, this shouldn't be I am, but I am. I guess that was a typo. And never used it before is expected. So I think this is to what I expected. Thank you, Dan, coming to rescue.
All right, we should be back.
Yes. And can you see my screen now?
Yeah.
All right. So how do we go now capture this information in these things. So these are the guidelines. These are MSI. I sort of hinted towards what MSI is. That these are a set of guidelines on different aspects. So I cheated a bit. So you should have also said I've heard about it because I already mentioned these are metabolomic standard, minimum information, and guidelines. But going back into how can we make our data into a digital format, we use lab books, or most people use lab books. And they go back to the lab book trying to find it. It is the most extensively used, but these are not machine readables. The machines cannot do anything with your lab book. You need a medium, need a format that could be machine readable. The ones that we use and promote, for example, for metabolite data sharing is called ISA. It goes into the details of it. It stands for investigation, being the top layer. This is what I'm trying to investigate, a disease, a phenomenon, an environmental study, and I set up a set of different studies. One of them could be a metabolomic study, could be other omics studies, could be proteomics. And then I do a set of assays. So this is what ISA stands for. An assay could be your LCMS, GCMS, different type of LCMS, negative, positive. Eventually you create a data. So we can actually capture all this thought process in a pipeline. And what we do with it, once this data is captured in digital format, they could be tagged. That makes sort of a control vocabulary, makes these information machine readable, so machine could understand what you mean by certain terms, because it's very different the way the language are spoken and the way computer understands. Our spoken language has to be far more clarified. Then once they have this information into a digital format, then it could be relationships and networks be used to make more sense. This is where the computers come to the aid of data to provide a meaning for it. You probably used Amazon for buying. He always says, oh, people who bought this, they have this similar shopping habit. So this is what information is being used from your shopping habit to provide suggestions. A similar thing could be done biology. These are known, for example, as RDF statements. I'm not going to go through there, but I will go more details on how do we do it in a control vocabulary format. Again, this is my analogy of standards, trying to keep everyone engaged. This is a train track, gauges. It's the length that the gauges or rail tracks are. And as you can see, the trains can flow in certain continents or certain places a lot better, and they get stuck when they go pass through one standard to another. So this is sort of the analogy why we need certain standards. We could have more than one standard. There's nothing for it, but too much of it could be also a hindrance. But if you work and find the best one, the most suitable one, essentially, analogy being that data flows a lot better, the same way a train can reach far distances, the more the standards are adhered to. Now, a bit more details. So what it is ISA mean, I already mentioned. You can here clearly see the investigation, study, and assay part, the relationship to it. So for the sample, we need sort of information. These are the MSI guidelines. These are the minimum information. Well, we always say that we actually done a minimum or minimal information because that minimum information was even too much. When you ask the people to report all these details, they said, oh, this is too much. That's why I went back to the poll. We do not want to get to a month or 2 months. We want to keep it reasonable. And so you could be engaged with data sharing. So if you see a week is acceptable, for example, on average, then you need to have this information provided. But these are essential information. So when you're reporting, you need to tell what species it is, because without that information is not usable for me if I don't know which species or which part of species for use. There are other metadata information. So they're all being captured in the ISATAB format developed here. If you go to isatools.org, this is a group in Oxford eResearch, who maintain and develop these tools, and their whole team of which we are actively collaborating. So the format will provide a workflow, a sort of a guideline that you go in the same way you follow your paper. You make your title, you make your abstract, you write your materials and method, and then you go through results. So it provides the same process. So you could follow. If I break down this process, it kind of look like that. It might look a bit overwhelming, but it provides the current platform called ISA Creator, which generates ISATAB format. It's a Java application based, so it should be platform independent. It provides a similar spreadsheet looking like templates and formats that people are familiar with, so they can copy-paste the sample name, and metadata generate to it. There are tools that you can link it to the publication, or actually you can, once you insert something like factors, which are important for your metadata, for example, you got a group of samples, there are the ones that are healthy, there are disease, there are male and female, so these are your factors. These factors could also be visualized. And also, the interesting part is that these also could be linked to a sort of control vocabulary or ontology that you can use to make these data now machine understandable, as well as at the end linking the raw files. Individual raw files came out of instruments to all the metadata for it. I know this might be too complex and it's the first time you're hearing it. It seems overwhelming, but you get used to it and you will see, hopefully, there are better ways to generate these things. So if you bear with me right to the end and don't give up halfway through, you will see that we are, the ISA team and other teams are working to make this even more easier. So these are examples. You get study samples. You infer information about characteristic tag. For example, this was a mouse, where the mouse came from, what sources were used. You might have plant, for example, and you might use leaf of the same plant or root of the same plant, but the source is the same plant. So you need to be able to capture the information. So people who are really using the data, they know, the leaves, the roots, and the flowers, for example, came from the same tree or same apple or the same plant. So it is a source to be traceable, as well as more details of parts and protocols. There are also information tagged about protocols and parameters that you can say, what was my setting or protocols I used, what extraction I used, what was the type of solvent I used. All these related to how you handle or deal with the sample. So all I'm here talking about is, at the moment, about samples. And then you have factors. I give an example about factors. For example, they say strain A will be the genotype factor. Or if there's a diet related, that will be another factor. Maybe the high-fat diet, low-fat diet, or days could be done. But these also need to be controlled as well. You're not saying a number 30 is not enough. You have to tell me, what is that? Is it 30 days, 30 hours, 30 minutes, 30 seconds? So these need to be carried, or we need to have this unit as well. I guess for assays, it's a lot easier. You're more familiar, so I'm not going to dwell here. If you do a different assay, you know what to report. This is essentially your materials and method. What was my instrument parameters or settings or column settings that I used? So they're all places to capture the same information. How we derive these things? By a configuration file. So configuration file is in a way to help, because there was one of the working groups of the original MSI was the biological context. So they classify things into in vitro study, clinical study, plant study, toxicology study, to different type of reporting. So this type of reporting, depending on the context that you're working on, if you work in a plant or you work in mammalian, you must have different reporting type of configuration files that ask the right questions from you. So this is our bottleneck at the moment. We try to populate these configuration files to make it easier for people to say, I'm working, this is a clinical metabolomics. So I need to report what is relevant if I use the clinical guidelines for populating. I mentioned ontology and control vocabulary quite a lot. So what is this? What do you mean by that? So if I give an example, a simple term called cell. If I say a cell, you might not know exactly what I'm doing, but if I bring it into a context and say I use my cell phone, the cell, to contact someone, or I use I did an experiment using cells from this species, then you know what I'm talking about. But for computers, this is not possible. It could have different meanings, so we need to find a means or terms to make them defined. So we define all these terms, what they mean in terms of databases and resources. This whole group of people, a lot of people working on ontology, providing a means of capturing what's done in the experiment in a controlled vocabulary platform. So then you can look it up and say, this is the term I meant as cell, matches this description. So I'm going to choose, for example, a prison cell, because that's what I meant in the context. The same thing happens. The computer, the software will look up and find you. So for example, this is how the example lookup looks like in the isotype tool. I must say I'm looking for C. elegans, and then it looks it up, does a search, and provides hit list. Among the hit list, you go and choose, I'm looking for this particular species. Once you capture this information, it gets all the information relevant to taxonomy and some kind of ID. OBO ID, which is very well known ontology platform. So then computers can say, I know what you mean. So this is taxonomy. It has related synonymous. It has this label. It has this information and a cross-reference. So if I use different resources, they all know what I'm talking about because we're all talking about C. elegans. So once you go through the whole process of capturing this information, what do you do with it? The next step will be you going upload it. You upload it into Metabolites, and you will see it. In Metabolites, once you upload it, there are two ways in Metabolite that you can browse. I'm going to go a bit faster here to catch up with time. You can always go to Metabolite and do it yourself. In there, you provide information based on studies and based on compound. So browse studies provide all the metabolomics experimental data, all the files, formats, information that you captured in isotype format now presented there. And in browse compound, it will be a compound-centric, metabolite-centric. In there, you might have information about pathways, reactions, or reference compound like MSMS or different types of mass spec data or NMR data. Again, there are a lot of resources similar to this exist. I'm sure you're all familiar. I'm not going to go there, and there's plenty out there to go by. Once the data is uploaded, it goes into some sort of a checklist, a validation. This is a traffic light validation. The green is all validated, all good. You provide all the new information, and it goes into amber and red, which are the missing information. So this is an example of study validation. It checks, do you provide title? Do you have this description? If it's not, what's missing, it will be reported back. So this would be a good way, a guideline for you to go and back say, okay, I need to provide that. Or you get it back in touch. Once in the curation process, we're moving now here from submission to curation, and you say, do I need this? But I don't have that. So curator will work with you, provide this information, structure this information, so for it to pass validation. After passing the validation, it could be in a review process. That review process meaning that you can now share this data. It's ready to go public. You can share the link for a journal. So by default, when you submit, everything is private. Nothing is public. It looks kind of like this. You will get a unique ID or accession number starting from MTBLS and a number. This is when the data is public, this is accessible. But once it's a review process, you can ask for a private shared data. So provide an encrypted access to the data set. Also the information you provide that you can share with your colleagues, collaborators, or if you want to give access to the reviewer of journals to look for it. Also the data here, you can see all the data in the study files and all the files in both an open source format, a commercial format are there. Anything that you want to share, we do accept. You can share any type of data sets that is relevant or information required. So we don't have any limitation on our side. Obviously to make it work, you need to provide a lot of data. Your QCs, your blanks, your raw files, your reference spectra that you use in-house for ID. There isn't any limitation from our side. We do accept, and we would like you to share as much as possible. Challenges are metabolite ID. Always metabolite ID is challenging. I mean, this is the most difficult part that people struggle to this for metabolomics. So we do a lot of curation steps. We try to say what metabolite reported, what exactly you meant. You have to find a database match for it. This makes it, again, machine readable and understandable what exactly metabolite you're talking about, because there might be some variation or differences between metabolites that you haven't considered. Again, this is an area which is quite hard. It needs a lot of work, and it is a community work to be able to provide a guidelines and a means of reliability. Like this identification is done properly, or is it just annotated, or is it just a database match? So I've just looked it up and I've annotated, but I haven't ID'd. Or actually, I have more evidence now. I can say I've done a lot more. And I can be proven that I've done a further more work in able to identify and provide backup data for my identification. So this is, again, a long story. But once it's there, it will capture all this information that we can browse per studies and go and see what metabolite, for example, they identify. In terms of reliability, so we do this is a very tab separated format. What I forgot to mention that this ISA tab, the tab part of it, of the ISA, meaning there's a tab separated format. So it's a very simple format in a way that tab separated format is easier to use. A lot of people have done studies on data formats, and they found that the text-based simple studies as the file structure is the best way to be able to share this data in the long term. We're talking about 10 years, 20 years, 50 years from now. A lot of the formats might not be used or you can't read them, but the text tab separated still will be readable and accessible. So the simpler, the better. For that, you provide all the information about the database ID, some sort of characteristic, mass over charge or retention time, also a reliability score. This is based on existing 2007 guidelines, that if it's a level 4, meaning that you just you don't know really what it is, you have some characteristic information from M over Z, so you annotate it, you just match them. Could be any of these 20, 30 compounds. But you might have more information, you know the class of compound. You know, I need a bit more. I know what group belongs to. I don't know exactly which one, but I can narrow it down. And if you go against more details, you can increase the levels to level 1, which is like of enough evidential supporting experiment or data or metadata to do it. Again, these are constantly being looked at. They're also being reviewed, and there is a task group to look into these things and find the better ways, the better means to do it. I guess this still remains the challenging part of metabolites. So moving to the data formats, I mentioned this, the simpler the better. I don't know how many of you can recognize these things. I wish there was a poll or a quiz. I could say how many people can recognize what, for example, this one is. Or do you have these things in your house, a mini DV or Hi8 or Digital 8 or VHS or Betamax from your childhood videos and families that you don't know how to read it anymore? These things happens all the time. It will be over time, you will not be able to access and read some of these formats because technology changes. So we need to have a means to capture this information to avoid loss of data. So this goes back to this coordination standard, Cosmos. There's a group of people involved in a big partnership across Europe and worldwide. This is not exclusive to Europe. It was the funding came for it, but it includes partners from around the world to be able to say, let's work on a format that it has a more long-term support. Most of these formats are called XML. So XML, what is XML? This is what XML stands for, Extensible Markup Language. It is basically a set of language, a format similar to HTML. So if you look at your browser and look behind your web page, you see all this code. All these codes or tags have a meaning that what exactly I'm trying to do, which this is a title, this is a subtitle, this is the main text. So in a similar way, you provide tags. This is my instrument, this is my probe, and you tag this information in this list of information relevant to the data, and then you bundle up the raw files to it. So these are a medium which are neutral to the vendor format. If the vendors provide support for open source XML format, you can convert them into the open access format. So then you don't need a specialized software to read it. And hopefully there's a more future long-term support. The analogy to the disk and file format you saw. So there were essentially a format to carry data, not necessarily to display it, but the data part was to it. There are continuously work on it. This works mainly started from, if I skip this slide, you can see that it's been there for a long time, from 2006. While we didn't have any metabolomics database or resources, they had proteomics resources and already worked on evolution of these formats coming to an N7ML, which is sort of the mature format that is commonly used in proteomics and metabolomics, that a lot of vendors provide libraries to convert their raw format into open access format. Another format is MZ-Tab, which is a tab separated. This is sort of a reporting metabolite identification. So this is our notion, idea of Cosmos coming from. This is sort of Stefan Neumann's work that you could capture all this metadata into this ISA-Tab format, a raw format in sort of XML format, and metabolites identified in a tab separated format. So then we have a complete picture of the whole process, from your metadata to raw files and to metabolites that are identified. Similar thing also could be done for NMR. We also, people out of consortium, the long list of people down here at the bottom, worked on providing a similar outline in an XML format for NMR. So be able to capture this information. Maybe this makes more sense. If you heard that there was an instrument manufacturer called Variant, now Agilent bought it later and then they stopped supporting it. So the support, they do still support it, but you never know, the support will continue in 5 years, in 10 years from now on. But if you captured it in open source format, the data always remains accessible, even though the manufacturer might not be there anymore or not supporting it. So now, how can we make this easier? Still, sitting down doing a month of work, no one wants to do that. No one wants to sit and capture a lot of this data information. So the best approach for us, for you, for everyone, is to have tools which help with these things. So if you're lucky enough and you have lab information management system that you run and control your experiments using a digital format already, for example, this is Metabolomics Australia LIMS system. This is working, which is coordinated by Saravanan, and he's the project lead on these things for Metabolomics Australia LIMS system. They're working and they're very to provide a means of exporting this data. So once you have already a system in place, you capture this information, you can generate ISO file. You're also working with other partners as well. This is what now, if anyone is there, I will really like to get people involved, engaged in providing support. We have a very nice collaboration with Biocrates that using their kit, you can actually generate ISOTAB format for all the metadata that already gone through the process, for even your metabolites that you identified. If you use NZMine 2, the new version of NZMine, for example, will support an ISOTAB format. There's also a module, if you use R, if you like R for data analysis, you can use RISA. Also, there is one for Galaxy. So the more and more the numbers are joining to be able to produce, and there's a lot of people who haven't mentioned. The recent is Bruker's Metabospace, which is the platform providing to run and do some analysis. And there you can see they're also now gradually supporting. So the idea being that the more people who join, more software people who make already this software, a lot of this information is already in your tools, in your softwares, in your formats. So we work with Birmingham University as well. There's a PhD student, Tom Lawson, and Ralph also here on an EMN. They together are reading all this data from your XML file, from your raw files, and generating an assay-based ISOTAB format. So there is a lot of scattered work if you try to get together to be able to use it. If you're a computer programmer, and this might be alien to most of you, then there is a programmatic access to be able to generate ISO as well. So those of you who are programmer interested, go and look up the GitHub. Those who are biologists don't care, skip this one. So we're going back still. Always motivation to data sharing is hardest, but we have a good example. First, as a carrot, why do I go share my data? Who cares? I can still publish my data with it. Well, for now, yes. But maybe more and more journals are asking, no, actually, if you want to publish in here, then you need to share your data. If you use a public grant or funding, then you might be asked to go and make your data available because you use a public source of money, so you have to make your data available. So this cases of getting narrower and narrower. Also, it's a good practice. If I share my data, it increases my citation. People can go and make a tool and reference to my paper. So I will have a better citation. These are two examples that I picked up that are use of metabolomics data. Fred Brett, Jan Strandrup, which is a fantastic guy. He's helping a lot in EMN, as involved in setting up the forum. He also went and used a lot of data sets to improve his algorithms to predict retention time. There are also a group from Roy Goodacre went and they want to test a missing value substitute for multivariate analysis. They used a lot of data in metabolites to do it. So there's a cases of going and reusing. And if you have gone and reused, please get in touch and let me know so I can add you to the list of people who went and reused data. That would be fantastic. So, almost finished. The last part, what is the missing link? I'm going a bit fast here. I know I'm running a bit of time. You probably are already exhausted and tired. Keeping you on this subject or standard is not as exciting as many subjects could be, but I'm hoping to get you engaged. So this goes back to tools. Rachel, our PhD student, has actually collected these things because she wants to know how many tools are there. So keep finding tools and doing that, and this is the perfect table illustrate the plethora of tools available. And when the total number came to 208, I really couldn't believe that this many tools exist, and these are open source tools. They're not commercial. They are non-commercial. They're not tools that it's mostly an open source tools. Mostly tools that are created in academic or labs. These are non-commercial tools, and 208 is quite a lot. And about 93 different tools and routines and algorithms for CMS. So which one should I use? Obviously, it's a nightmare. There's so many tools to do. And if you use tools, you will find that it's not actually the path that I take to analyze the data. This is a very nice paper by Jennifer, Ralph, David, Birdhouse, and Mark. Please go and read it in Scientific Data about direct infusion, and they provide all means of pathways, routes that they can analyze the data to get results. So if you do a bit variation of these methods and this route, you might not be able to get the same result. This sits in MTBLS 79, that's the ID, and you go there, you have all these formats in between, all those one out of this pipeline produced, so you'll be able to reproduce the results of the same experiment. So I can do that. As you all know, if you took a different route, you change a parameter, your result might change. This is only on data processing. We're not talking about, we're not going even talking about different instruments and different acquisitions. That's a completely different ballgame. I stick to the data side of stuff rather than going into the analytical part, which is by itself a lot of challenging. So generally, the choice of tool, the tools that you can do, tools that can provide a traceability, an audit, something that you can reproduce. So there are examples of it. The example of, for example, 9 is one example. Again, this was done by one of the worker PhD students at EBI. He liked quite LCMS data analysis, so he used a tool called Mass Cascade, which you can actually go and input the raw file, the example file, and do different analysis process, from creating feature, removing baseline, removing noise, and generating end results. What he would do is that if you share this workflow with someone, if I share this workflow with you and give my raw file, you will be able to, at least, if you're lucky enough, going back to the example of even the perfect digital medium is not 100% reproducible, you put your raw file, you might get exact same results. And this is great because now you can go and share this result. Similar thing again done in Galaxy M, which is another Galaxy medium to capture this workflow. So what the thing is, the workflow does, I want to emphasize here that a lot of them, for example, the Galaxy here, done again by authors down here, I keep mentioning Ralph, so you're partly responsible for this talk as well, Ralph, is that uses existing tools. So we're not adding to the complexity of this big table yet another tool. But these workflows use existing tools, whether they're in Python or R or MATLAB, and provide a framework. This framework makes you to go and create a pipeline so you can generate a reproducible result. They have boxes. This example provided by Galaxy. You can put relationships. I get my input, do certain tasks, take it to the next steps, do several tasks, do it, change the parameters. You can always see what parameters used, and you can even share these workflows. So I can take someone's workflow, improve it, and do it. So for metabolomics, one of the best ones for now is workflow for metabolomics. This is by a consortium of a French research group that is on the MetaboHub, and there's a long list of people collaborating, providing different tools, and you can see the plethora tools that you can use for CMS, for GCMS, for NMR, mass spec part, and even statistical part to be able to go and analyze your data and generate, even up to annotating results and generate tools. So we did, with the help of Etienne and people working in his lab, in CEA, we went and used Metabolites, for example. We use MTBLS1 as an example, an MTBLS2 and 3 as an example and went through the whole process of now you have the raw files, now you have the pipeline, now you can go and generate using this pipeline to produce exact or similar results once we did all the parameters right to the original paper. So this will be a now better chance of providing data, providing a paper, providing a research, providing raw files, and all the data for you to go and not just read the paper, but analyze the result and get everything out of it. All of these ideas also could be go back if you have more time and you're missing something. I wasn't good explaining it. You can go and read this. Data standard can boost metabolomic research, published open access, so you should have no problem accessing it. There's a direct link here. I will share this results in the slides, in slide share, so you can actually go there and have these links or contact me or any of the authors or use Google as your friend to be able to pick this up and read more details are part of it and get involved. The more people involved in the whole process, the better. This is a work of a lot of people. This is ISATAP team members. For the time, I'm not going to mention all the names of people. A metabolite team, which help with the curation, software engineering, different aspects of providing services, sharing, curation, all this data. The consortium of people who work on different standards in metabolomics, the PSI community, also here, and a lot of other people who are involved in this aspect, and the new phenomenon, which is continuation of a lot of these standards, which I haven't talked about at all, are funders. And I quite like this, the last message that you share your data, make it available. Don't be worried about if it's wrong, then that's good. Be prepared to be wrong. Otherwise, you will not come with something original. So I quite like this quotation. And again, thank you guys for bearing this long and staying and listening to me. Post your questions. I can't see any.
Hi, Reza. Thanks very much. Baljit again. Thank you so much for an amazing talk, and really for you to walk through with us the amazing tools that have been developed over the last years for metabolomics. So yeah, I have a bunch of questions that have been coming through. We've got about 5 minutes. We're going to run a little bit over, but I hope people can stay on the phone so we can address some of your questions. So one of the questions I had here, somebody just needed to clarify the fact that, do I need to convert my data to XML in order to upload my data to metabolites?
No, but it's a good practice. We have good practices, and it is not mandatory. Some of them you can't convert because the library is not available. But if available, you can do, but it's not an option, a mandatory option. It's considered a good practice.
Okay, and what about NMR data? Could you load NMR data up in here, or is it just mass spectral data?
No, you can do any data. You can do imaging. We have data set from imaging. We have Raman. We have direct infusion. We have GCMS data. So it's no limitation of type of data. You might be not finding the right configuration file, so we need to improve our configuration file to work, for example, for imaging data. We're working with Theodore Alexander Group that they do a lot of imaging, and they have a consortium of these things to try to get a lot of the imaging parameters included, so the people who use different type of imaging can actually report there based on their guidelines. We work with the environmental group. A lot of people who've done fantastic work in reporting metabolites from oceanographic or environmental. The needs are not necessarily could easily be found. There are some guidelines, so we try to work with them, generate the configuration files to be able to capture those things in terms of experiment. But both in terms of type of experiments and the type of assays, meaning type of experiment, if there is anything missing is our lack of time and resources to be able to capture that type of experiments, but there isn't any limitation.
Sure. And what about, so right now you addressed metabolomics applications, lipidomics applications, the questions come in, does this apply to peptides? Another type of data, I mean, how do you see this working with, say, for example, is it part of the Metabolomics Standards Initiative to start trying to integrate data with, for example, proteomics data you talked about, but genomics, further down the line genomics data as well? Do you see that part of developing here?
Right. It is a good question and a difficult question. This is our overall aim of EBI. EBI is not me, it's about 600 other people, and there are important people who make bigger decisions, the directors and others who have this in mind. So we are working at our back end on a project aptly, nicely named glue, to be able to glue different data. It's not something that you can use on a broader sense, but this is the idea, to be able to use these resources, cross resources. So this is sort of planned, but these are very much early work. The challenge of it is quite difficult to be able to scientifically and data and understanding cross-platform is very, very difficult, so we need to work with a lot of people to understand. I don't understand anything on transcriptome or proteome or that, so I need to work with people to say how we can integrate these things. But these are pretty much early work in progress.
Yeah, sure. So I've got actually a question of my own that I'd like to put to you, Reza, in relation to the previous question. I don't know if you've heard of an initiative called Chorus. It's sort of led by predominantly the proteomics community, and it's come from Mike McCoss's lab and Nathan Yates at University of Pittsburgh. And Chorus is very similar to what you presented in terms of data repository, but actually not just incorporating proteomics data. Most of their users, of course, where they started out in loading up proteomics data, but they've now opened it up to metabolomics, lipidomics. They've got genomics in there, imaging, like you mentioned, HTS. And so the idea is to start integrating the omics. And I wonder if that's something that, next time you meet with your team and the rest of the group at EBI, it would be to perhaps how do your tools fit in relation to something like Chorus, and would you guys want to be involved in making these data more integral?
Right. I mean, there are several of these similar initiatives that overall EBI and us and others are involved, and this is really good. I think this is what a lot of people say. This is not some original idea come up with during this talk or another. This is a lot of people come up with similar ideas. So there are a lot of initiatives and fundings going into different medium platforms, disease-related, topic-related, subject-related, to be able to capture all this information relevant to a particular field, disease, or environmental aspect, and then be able to integrate this data. So I guess everything goes by the same notion that the funding comes and people get interested into that, and there will be research done. Some work will succeed, some will probably less successful, but I guess this is sort of a natural progress of things that people are interested in taking next step. I think there are a lot of things that we need to fix in metabolomics first. I will be less ambitious and try to fix the things that need fixing in immediate. And if you get these things right, and if it then integration in future gets a lot easier. These cogs and wheels had to fit very nicely together. If the one is round and the other one is a triangle, they will not really fit together, and then you have to go and reinvent these things. So it would be nice to actually work on these standards. That's my analogy for train tracks. If you have these things nicely worked out, then these flow of information in future, whether that's integration or cross-platform, will be a lot easier to have these tracks at least understandable, workable, and fit together. So we do that on a lot of parts. We even do, for example, test data exchange between Workbench. People at Metabolomics Workbench are great people who we collaborate quite closely. So we really like people to share the data, wherever there is, either Metabolomics Workbench or Metabolites. We will get around that by actually talking to one another and be able to do a data exchange. So there's a lot of data exchange practices we do and try to see where things fail and where it works, where it doesn't. So these data exchanges helps cross metabolomic resources and beyond metabolic resources, the other omics to be able to fit things together. But then again, these are very early on.
Sure. Going to sort of do one or two more last questions before we wrap this up. Very interesting question that's come in that's in relation to the slide that you have up on the screen right now, Reza. What type of QCing is done of data that's being uploaded into these repositories? How are they controlled?
This is a very good question, and I think we think about this a lot. It's an evolutionary process for us. So things we like everything else, we learn, we change, we experiment. So it's not like people who work in databases and resources, they figured everything out. They haven't figured anything out, but they're equally learning as well. So the things that we did early on and the things we're doing that, it's very different. So we try to increase the quality. Our first challenges were just encouraging people to share the data. So if we put very stringent criteria, no one would have shared. So we were in 2012, 13, it was called begging, going asking people to please go share your data. Come on, we could help and this and that. We're getting to a point that now more people are aware and are willing to share the data. So we try to tune these things together to increase the quality. We are at the stage that a lot of these can be adverse. So if there is a shortcoming from our side, the data sets are not that big. We've got about 300 data sets together with Workbench, maybe there are 500 or 600, still is fixable. But what we didn't do intentionally, we're going to check the data as well. So we thought this won't be our job. We don't want to take this role of checking people's data and accepting on the basis of quality. We leave that to reviewers. So if your data goes into a journal will have reviewers. If reviewers go and check it, we'll do it. Another checkpoint would be if people start to use the data, to share the data, that feedback mechanism could be captured. Again, these are not there yet. But it could be done. Other people have done this very successfully in San Diego group, in a massive database. They're using this feedback mechanism quite nicely as well to be able to get the feedback mechanism. So short answer, we have not yet implemented a quality check. Our checks and validation are in terms of numbers. You provide all these criteria that we need for reporting. But whether that experiment is a good experiment or a bad experiment or QCs are done properly or they haven't done properly, that we do not do. We leave that for the reviewers. What we do ask more and more now, becoming more and strictly, say, well, can you provide your QCs as well if you're sharing data? Because if you give your data but don't give your QCs, then that data is not very usable for other people. So we will do that. We were not as strict before, but gradually, as people are being more aware and using it, that strict criteria of asking for a more, or your run order is really important. If I don't have your run order, there are ways to get that. That will be very, very difficult to see exactly what's been done to get it reproduced. Again, I think the analysis part, the missing chain in the link, if it's a workflow process, that also will fill a lot of these gaps. So if I go through the work process, all the pieces of a puzzle of the raw files, the QCs, will fit into the workflow to be able to run them. Then I will have a very nice complete picture. But again, we are learning.
Yeah, absolutely.
Very long answer.
Thank you. So last question to wrap up this webinar. And I think this point has been made before, how metabolomics has sort of learned and taken from proteomics, which is why we're so far ahead in a quicker time space from where proteomics was. But so how close are we now to metabolomics reproducibility? So really answering your question, answering the title of your talk.
I got caught on my title. Thank you. Very far. The answer is yes, very far. Yes, very far. Again, I tried to briefly mention that. Then what I'm talking about is only on the data side. I'm not talking about on your analytical platform. I'll assume that are all done the way that it's been reproduced. The question of biology remains the question of biology. This is very, very challenging. Mm-hmm. Question of result. Every measurement has error. So no single measurement is perfect. You always have certain error measurements. and these things exist in data acquisition. And not every two instruments, even by the same manufacturer, are 100% identical. There are electronic tiny bit differences. So there are, but the question of biology measurement remains the same. So maybe if you're imagining you're measuring on an average measurements, they should be right. Maybe someone thinks that some of them are, some people are taller, some people are shorter, but the bigger biological questions should remain the same. There are ring trials and experiments. A lot of people are doing it. What I'm trying to stick to, so forget about this analytical part, on the data side. Once the data is reproduced, then that's a little bit easier. Still not 100%. If I share these workflows and ideas, and if I provide all the steps I took, theoretically, again speaking, you should be very nice. The two examples we try to work on with people at CEA and other people who in Metabolomics workflow, to be able to provide the data and from the data to get the same end results. Obviously, there are so many cases. The thing is, Metabolomics sits in the cross junctions of informatics, in terms of chemistry, biology, and analytical chemistry. So it's just very, very difficult, very, very exciting as well. This is one of the very few sciences you have to know quite a lot of stuff to be able to do, for example, metabolomic experiments rather than a single one particular field, for example, statistics. You should work with a lot of stuff, so the challenges are a lot bigger as well. So it's a kind of a butterfly effect. A small errors from this and there will end up being significant differences. So to control all of these things is very, very difficult and very, very hard. Hence, going back, yes, reproducibility so far is very low. But if you work gradually towards it, and others have done that, I have to say, genetics people and others are very, very close to creating this reproducible results, at least in a certain aspect. So I think metabolomics will reach that. It's just an evolutionary process.
Thank you, Reza. So on behalf of the EMN, I'd like to say thank you again for presenting today. It was a phenomenal webinar, and at least to some of our early members on the phone and on the WebEx, good for them to see what's being done on not the analytical side, but once that data has now been acquired and analyzed data sets. So thank you once again. Look out for our next webinar, which will be in April. Keep posted and follow us on Facebook and Twitter and LinkedIn, and we'll see you next time. Thanks again, Reza. Thank you to everybody.
Thank you very much, and please participate and share and join. The more you people share and join, the better things get. Thank you again. Excellent. Guys, for organizing these things. A lot of hard work and setting up, so thank you guys as well.
Thanks again.